Good evening, everyone. Welcome to week 12 of Living Systems 1. And we are really on the home stretch now with 2 lectures to go. Tonight we'll be talking about taxonomy in lecture 17. And then we'll also look at the first half of the final lecture, lecture 18, which we'll finish off next week. So we're going to have a very brief look at classification of organisms. And then classification, of course, involves grouping organisms into particular categories, and those organisms will share common characters. So let's think about the function or the role of classification. First, classification aids in communication. So it's much as mammals or dicots or monocots without having to run through all the characteristics that those particular organisms actually possess. A second important function of classification is that in the storage and retrieval of information. So, and also another important function of classification. Classification allows people to predict information. And there's a nice example here from your textbook. So what we're looking at here, these are the seeds of the Moreton Bay chestnut tree. And what scientists discovered was that a particular compound within these seeds, an alkaloid, actually possessed properties that inhibit cancer cells in test rats, and also inhibits one of the proteins that's produced by the AIDS virus. So in order to search for other sources of this chemical, this important chemical, they looked at the characters found within this particular tree, and in particular they looked at the morphology of the pollen that it produces. So in other words, the form, the structure, the size, and so on of the pollen. And then they looked for other trees that also produced pollen with similar form and structure and size and colour and so on. And they identified this tree here, which is found in South America, and sure enough, it possessed exactly the same compound. So predicting new information is also an important function of classification. All right, so classification is studied by biologists called evolutionary biologists. And what's very important to them is to classify organisms according to phylogeny. This means that that organisms are grouped together into categories according to a pattern of common ancestry. And the study of phylogeny is referred to as systematics. Classification is dynamic. It changes all the time as additional information about relationships becomes available. And this is very much driven by advances in techniques that scientists use in classification. So with the beginning of the era of molecular biology, all these new techniques, particularly dealing with DNA and RNA and proteins, enabled classification to become even more specific. And with those techniques, they ended up changing the way in which a lot of organisms are classified. All right, everyone's familiar with a tree of life. Just like the way that you would construct your family tree, scientists can construct these trees of life, and they're called phylogenetic trees or cladograms. So here we have an example of a phylogenetic tree, although to me it looks like it's a tree that's fallen over. This particular tree is an unrooted tree, so it's not actually showing that primitive ancestor that diverged over time to give rise to all the different species. But nevertheless, it still gives some valuable information. Let's have a look at it in a little bit of detail. So this branch here obviously represents the land plants. In yellow, all the yellow branches belong to this group called the protists. Over here in red, these are our prokaryotes, here referred to as the monorans. And then down here, we've got another 3 branches of the protists. This one here is the fungi, and in blue, we have a couple of branches representing the animals. So the way that we interpret this is to look at how close the branches are to each other. So if we look at this one here, this branch representing the land plants, the closest branch to this one is the green algae. So of course the green algae are said to be the closest ancestors, or closest relatives rather, to the land plants because of the proximity of those branches to each other. Another example down here, if we look at the animal branches in blue, and only just 3 examples are shown here. The closest branch to the animals is this yellow branch. This is a group of protists called choanoflagellates. These are unicellular aquatic organisms. Our aquaculture students might be familiar with these. And they're positioned here very close to the animals because they share a lot of gene sequences, a lot of similarity in their gene sequences to the animals. And in fact, that's how this particular phylogenetic tree was constructed. What they did, they took ribosomal RNA genes. So this goes back to last week, once again, ribosomal RNA genes. They actually looked at the nucleotide sequences within those genes and then compared them across all these groups of organisms. And the closer the sequence similarity between those gene sequences, then the closer different groups are said to be in terms of relationships. Through an evolutionary timescale, that is. So let's look just briefly at how this works. If we had a group of organisms, we'll call it group 1, and they had a sequence of nucleotides in their ribosomal RNA gene, A, C, G, T, A, and I won't write all the other nucleotides in the gene because it'll take me too long. Okay. And then they take the nucleotide sequence of ribosomal RNA gene from another group of organisms, and they find that the sequence is A, C, G, T, T. Okay, there we go. Won't worry about the rest of it. You can clearly see there is a difference here in nucleotide sequence. So we can say that group number 2 shares 80% sequence similarity in their ribosomal RNA gene to group 1. Okay, so very similar. So, for example, if that was the case with these 2 groups, the green algae and the land plants, then they would be positioned very close to each other on this phylogenetic tree. If the sequence similarity was very low, if it was, say, 30% or 40%, then they would be much further away. Okay, so just to show you basically how that works. And I'll just clear that. All right, so just for your interest, well, particularly for aquaculture students, here's another example of a phylogenetic tree. This tree is actually a rooted tree. So we can see the roots down here, which shows that the ancestral species that gave rise to all these other species are the bony fishes. And then over a period of time, some of these bony fishes are said to have diverged. That is, they changed in terms of certain, they evolved certain characteristics that basically made them a different species to the original bony fishes. And these were the lobe-finned fishes. Over another period of time, there was another divergence event, and this resulted in evolution of the sturgeons, for example. Now, don't ask me what sturgeons are. I'm sure Darren and Brad could help us out there. And so on. So, of course, we're looking at this time frame here, which could represent millions of years in terms of evolutionary history. And right at the top of the tree here, we have the spiny finned fishes, which you can see were the most recent to have evolved.
Sturgeons are best known for their caviar.
Oh, really? Thank you very much. In that case, I do like the sturgeons. Okay, so the field of study that deals with methods and rules and principles of classification is called taxonomy, and these scientists are referred to as taxonomists. There are set rules for naming organisms. There are written codes for consistency. So there's a written code for naming plants, another one for naming animals, and so on. And these codes are referred to as international codes of nomenclature. Okay, these codes basically include all these different levels of classification and levels or ranks of classification. Each level of classification is referred to as a taxon or taxonomic level. With the highest level being kingdom, and this is the most inclusive. The lowest level, lowest taxon, is the species level. This is the least inclusive. All right, here's our taxonomic hierarchy. Kingdom, next level down, phylum, class, order, family, genus, species. As I said to the students this morning, this would be a very, very good slide to memorise. So think about some way of remembering the order of these taxonomic levels for the final exam. Okay, so come up with some way. I can't remember what other students have come up with because I do have my own method of remembering these. So let's just have a look at some of these terms. Phylum, so that's the singular. Where's my pencil? So plural would be phyla. There we go. Class, order, family, genus. This is an important one. So genus is singular. Plural for that. Plural is genera. So one genus, two or more genera. There we go. And then species is an odd one. The term species is used both in the singular and the plural. You might occasionally hear someone refer to species, one species. That's not generally accepted. Okay, so just stick to species and species, whether it's one species or two or more species. All right, there are some intermediate ranks sometimes indicated by the prefix sub. For example, subphylum would be in here, subfamily in here, and subspecies at the bottom there. Oh. Okay, clear that. There we go. All right, couple of examples for you. You don't have to remember these. Grapevine for our viticulture students belongs to the kingdom Plantae, obviously. Within the kingdom Plantae, there are approximately 12 or 13 phyla within this kingdom. The grapevine belongs to the phylum Magnolia Phyta, and all the flowering plants belong to this particular phylum. Class, Magnoliopsida, and this is where all the dicots are placed. So you might remember we mentioned the dicotyledons in one of our previous lectures. Order Vitalis, family Vitaceae, genus Vitis, and species Vitis vinifera. And an example for the students that are studying animals. The horse belongs to kingdom Animalia. Within kingdom Animalia, there are at least 30 different phyla. The horse belongs to this phylum, Chordata, class Mammalia, order Perissodactyla, which includes horses, tapirs, and rhinos, family Equidae, genus Equus, species Equus ferus, subspecies Equus ferus cabilis, which is the domestic horse. All right, so let's look at some important points on this slide. Or maybe we'll go back to the previous slide because there are no equine students here tonight. So let's look at this one. All right, first thing to note is the species name. There's always two words that makes up a species name. At every other taxonomic level, there's a single word. All right, next important point. The species name, or sorry, the first part of the species name is always taken from the name of the genus to which it belongs. Okay, next important point. Species names are always written in italics. So from second semester onwards, make sure that you observe this. Any taxonomic name, write it in italics. The first part of the species name is always capitalized. The second part of the species name is always lowercase. All right, so make sure that you continue this right through your degree, because once you get into third year, all these little bits and pieces that you learn along the way really start to matter in your writing. Okay, if you're handwriting an assignment, so for example, it might be a lab book that you're filling out that has to be submitted. Obviously, it's very difficult to write something in italics. In that case, if you're writing a species name, then you would underline both parts of the species name rather than writing it in italics. Okay, and we can see the same, exactly the same applies to animals. Okay, species name written in italics. All right, so there are codes of nomenclature, as we said earlier, and these codes of nomenclature set rules for naming ranks of taxa. So, for example, the name of the animal families always end in ide, and plant family names end in ace. Let's look at that. Here we go. Family name for animals, ide. As you can see there, go back again. Family name for plants normally ends in ace. Okay, just as a couple of examples for you. All taxonomic levels except the level of species have a single word name. So we saw that on the previous slides, and only species names are identified by two words. The first word is derived from the genus which that species belongs to, and the second word is referred to as the specific epithet. And this is also referred to as the binomial system. And the binomial system was first introduced in the 1700s by a very famous naturalist called Carl Linnaeus. All right, so we've seen that species names are composed of two parts, the genus name, which is always capitalized, and the specific epithet is always in lowercase. That combination of the genus and the specific epithet seen within a species name is unique for each individual species. So, for example, only the echidna is given the species name Tachyglossus aculeatus. Wedge-tailed eagle Aquila audax, and the rocket frog, Litoria nasuta. So we can tell straight away by looking at these species names that the echidna must belong to the genus Tachyglossus, and the wedge-tailed eagle belongs to the genus Aquila, and so on. All right, of course, there can be many species within the one genus. And an example is shown here, 3 different species of eagle, Aquila audax, Aquila chrysatos, and Aquila heliarca, all belonging to the same genus. And often the specific epithet is descriptive of that particular species. So chrysatos is derived from the ancient Greek, meaning golden, and there it is, golden eagle. The specific epithet itself may be used in different genera. So if we look at these examples, Litoria nasuta, the rocket frog, Parameles nasuta, bandicoot, Acropora nasuta, the coral. So 3 very unrelated species, but they all share the same specific epithet, nasuta. And that's because this is simply a descriptive term here, meaning nose. Rocket frog has an elongated nose. Here we have a long-nosed bandicoot, and a staghorn coral must have something that resembles a nose. I'm not too sure about that one. Probably should look that one up. All right, so what is a species? How do you define a species? What makes a species distinct from other species? It's the basic unit of taxonomy, the lowest taxonomic level, but it's not easily defined. Very, very difficult to define what constitutes a separate species. And for this reason, there are different concepts of how the species taxon is defined. Only 2 concepts are listed here. The first one is the biological species concept. And this is more commonly used by animal biologists because it's based on reproductive isolation between species. So in other words, if 2 groups of animals cannot interbreed, cannot successfully interbreed and produce viable offspring, then they're said to be separate species. Now, this is not easy. This concept is not easy to apply to plants. So plant biologists tend to use the morphological species concept, where they will differentiate different species based on phenotypic difference between them. That is based on characters which you can see, which you can measure, which you can observe. So for example, they'll be looking at form and colour and size and structure and so on. And there's other species concepts which we won't mention here. All right, so the kingdoms of life. The kingdom is the highest, most inclusive rank of classification. In the 1700s, the famous naturalist Carl Linnaeus classified all organisms into only 2 kingdoms. These were plants and animals. And there was a 3rd kingdom also for minerals. Later on, in the 1800s, a 3rd kingdom was introduced, and essentially anything that didn't fit into plant and animal kingdoms was placed in that 3rd kingdom. Eventually 5 kingdoms were introduced, and now 6 major kingdoms are recognised: the bacteria, archaea, protista, plantae, fungi, and animalia. Classification is changing all the time. Let's look at the different schemes of classification. So there are some groups of biologists that prefer this classification here. This is the 3 superkingdom classification, or 3 domains. Domain bacteria, archaea, they're our 2 groups of prokaryotes, and then all the eukaryotes are placed in the domain eukarya. Here we have the 5 kingdom system, All the prokaryotes are placed into kingdom Monera, and then we have separate kingdoms for eukaryotes: Protista, Plantae, Fungi, and Animalia. And finally, the six kingdom system, which is also commonly used. Bacteria, archaea, and then the four eukaryotic kingdoms. So let's look at some of the characteristics of each of these kingdoms. Bacteria, they're mostly unicellular organisms. They're prokaryotes, so of course their cells lack nuclei and membrane-bound organelles. Their cells are surrounded by distinctive cell walls. There are some autotrophs and some heterotrophs in this kingdom. So can anyone tell me just very basically how you would define an autotroph? Or a heterotroph? Or you can say no idea.
No idea.
Oh, that was easy. Anyone else? Does anyone know what's meant by an autotroph? No, you might not have heard this term.
I've only heard it on what's that show, the TV show.
Oh.
Yeah, but it's...
Oh, maybe David Attenborough. Did he mention it? No. Okay. Okay. So autotrophs, just in a nutshell, autotrophs are organisms that can basically make their own fuel molecules. For example, carbohydrates. Okay. Whereas a heterotroph can't synthesize their own fuel or energy molecules. They have to ingest them or eat other organisms that can make their own fuel molecules. All right, so I'll come back to these two terms, but let's move on to archaea. These are also unicellular prokaryotes. Of course, their cells lack nuclei and membrane-enclosed organelles. Excuse me. Their cells are surrounded also by cell walls, well, most of them anyway. There are some autotrophs in this group and some heterotrophs in this group. Now we move on to the eukaryotes, kingdom Protista. There are unicellular protists and very large multicellular protists, so a real mixture in this kingdom. They are eukaryotes, so their cells contain membrane-enclosed nuclei, and many of them also have membrane-bound organelles. Some of them have cell walls. There are some autotrophs and some heterotrophs in this kingdom. Kingdom Plantae, this includes the multicellular land plants. Their cells, of course, have nuclei and membrane-bound organelles. Cell walls contain cellulose. They are all autotrophs. So when you think about plants, think about photosynthesis, and the function of photosynthesis is to produce those energy molecules, those fuel molecules, in other words, carbohydrates. So plants don't have to ingest or absorb carbohydrates, they just make their own, and hence they're all autotrophs. Plants also contain complex organ systems. Moving on, the fungi. Fungi are mostly multicellular, but there are also, of course, lots of unicellular fungi as well, like the yeasts. Their cells also contain nuclei and membrane-bound organelles, of course, because they're eukaryotes. Their cell walls contain the molecule chitin instead of cellulose. And unlike plants, fungi are heterotrophs. They can't synthesize their own carbohydrates. They need to absorb carbohydrates, which they then use for energy. Kingdom Animalia, multicellular organisms. They are eukaryotes, so they have nuclei and organelles. Their cells lack cell walls. They are all heterotrophs by ingestion, which is a bit different to the fungi, which are heterotrophs by absorption, and animals also contain complex organ systems. So I think this is a really nice summary of some of those major characteristics of each group. All right, now let's visit each kingdom in turn and look at a little bit more detail. So let's start with prokaryotes. And the prokaryotes includes two groups, the bacteria and the archaea. So the bacteria, of course, are all prokaryotes. Characteristics, they are mostly single-celled. Their cells are surrounded by a semi-rigid wall. They lack membrane-bound organelles, and their genetic material, that is their chromosome or chromosomes, basically sits free in the cytosol of the cell, not enclosed within a nucleus. Okay, bacteria are incredibly diverse in terms of their habitat, in terms of how they function, and of course in terms of size. So let's just put that into perspective. So in blue we have a single cell of a Saccharomyces. This is a yeast species. It's actually quite small as far as eukaryotic cells go, but let's have a look at this. Diameter 8.6 microns. And of course it is still much bigger than our prokaryotic cells, which are shown here. This is a typical size bacterium, Escherichia coli, which has these dimensions, 2 by 0.5 microns. A smaller bacterium, Staphylococcus aureus, 0.8 microns. And here's a very small bacterium, Rickettsia rickettsii, with a diameter of 0.4 microns. But there are much, much smaller bacteria that have been discovered. All right, so quite small, but we can see these bacteria using our normal compound light microscope in the lab. Now let's compare those to viruses, even though we're not talking about viruses here. This is a large virus, a smallpox virus, influenza virus, the tobacco mosaic virus, and a small virus, the polio virus. And the size ranges from 0.3 microns down to 0.03 microns. Of course, these are too small to be seen with a microscope. We need a high-powered electron microscope to visualize those. Prokaryotes are divided into two groups on the basis of various biochemical characteristics. Superkingdom bacteria, formerly called the eubacteria, and superkingdom archaea, formerly referred to as ancient bacteria because they were thought to have evolved earlier than the bacteria. They now know that that's not true. The bacteria and the archaea appeared to have evolved at about the same time. All right, let's focus a bit on bacteria. Bacteria have incredibly diverse metabolic pathways, that is bacteria as a whole. And this allows bacteria to use most materials as sources of energy. There are only some plastics and organochlorine compounds that are resistant to bacterial degradation. So the OCs or OCPs, think of pesticides such as DDT, aldrin, dieldrin, heptachlor, chlordane, and so on. So they're chemical compounds that seem to be resistant to bacterial breakdown. All right, characteristics of bacteria. One of the main characteristics of bacteria is the presence of this compound called peptidoglycan, which is the major polymer found in the cell wall, in their cell wall. In the group known as bacteria, there are some photosynthetic species and some nitrifying species. We'll come back to that term in just a minute. All right, here's one group of bacteria, the cyanobacteria. These are the bacteria that can photosynthesize, just like plants. They're also very commonly known as blue-green algae, but they're not algae. This is just a misnomer. People see that blue-green growth in their dam or in their swimming pool and say, I've got algae growing. But these are not algae, they are bacteria. Excuse me. The reason for that colour that you see is because they produce a pigment called phycobilin. and this is a blue water-soluble pigment, which enables them to absorb certain wavelengths of light that their other photosynthetic pigments don't normally observe. So this forms that characteristic blue-green colour, which is more obvious when their population size increases. That is, they form those blooms in shallow waters. All right, another important group of bacteria can form very resistant dormant spores called endospores. They tend to form these spores in poor environmental conditions. So, for example, lack of moisture, lack of food, any sort of high stress environment, they'll start forming these spores. Here's an example. This is one bacterial cell and it's forming this endospore within the cell, and when that's completely formed, the bacterium itself will actually die and just leave that spore behind. All right, this is particularly important, the study of endospores, because some species of bacteria that form these spores, such as Clostridium tetani, which causes tetanus, Bacillus anthracis, which causes the disease called anthrax, are obviously fairly important pathogenic bacteria. Endospores are resistant to high temperatures, they're resistant to UV radiation, various chemicals, and so on. And they can lie dormant in the soil, for example, for decades. And then when the conditions are just right, they'll germinate, and there we have a new population of bacteria. All right, very briefly on the Archaeans, kingdom Archaea. Many of the Archaeans occur in extreme environments, including superheated deep sea volcanic vents and thermal pools. So these are really highly specialised prokaryotes, so they tend to not be as widespread as the bacteria in terms of habitats that they're found in. They are classified in various ways depending on the extreme environments that they do occupy. So, for example, some can inhabit super saline environments and they're referred to as halophiles. Others can survive in very, very low pH environments, even as low as pH 0, which is quite incredible. They're called acidophiles. Others can survive in really high temperature environments and they're called thermophiles. All right, characteristics of the Archaeans. Peptidoglycan is not found in their cell walls. So this is one of the biochemical differences between bacteria and Archaea. There are no photosynthetic Archaeans known as yet, and there are now nitrifying species of Archaeans that have been found, but this is still not reflected in the textbooks. There's often quite a lag time between when something's discovered and when it's incorporated into textbooks. So I need to update my that particular lecture slide. All right, let's look at bacterial populations on humans and in humans. Obviously, we harbour very large and dense bacterial populations, both on our skin and in our bodies. In fact, it's been estimated that if we were to look at the total number of bacterial cells that are occupying our bodies and the total number of our own cells, we would actually be more bacteria than we are humans. That's how many bacterial cells are found on us. Bacteria tend to cluster in moist areas, and the suite of bacterial species, which there are many, varies to some degree from one person to the next. There are quite a few different species that you'll find on and in every single human, but everyone does tend to have a few transient species that come and go, or species that some people have and not others. Really just depends on the person. These bacterial populations are incredibly important for many reasons, but one reason that I'll mention here is that the existing bacterial populations that we have actually prevent many attacking pathogenic species from gaining a foothold. So that competition provided by our own bacteria is incredibly important in terms of one of the first lines of defence to preventing those pathogens from gaining a foothold. All right, human feces. This is where you'll find an enormous number of bacteria, which are obviously derive from the small intestine and the large intestine, with the highest diversity of bacteria in the colon. Now, many bacteria are quite benign, but of course, there are some like good old golden staph, Staphylococcus aureus, that causes a range of different infections and diseases. There's another species of bacteria that causes the disease called leprosy, and this is called Mycobacterium. All right, let's look at some incredibly important bacteria and the roles that they play within the nitrogen cycle. So I'm not sure if any of you have actually been introduced to the cycles of the elements as they appear in any ecosystem. That ecosystem might be a very large tract of land. It could be a forest. It could be your own farm. It could be the ecosystem in your aquaculture pond, or even in your vineyard. But of course, there are different ways that the important elements enter these ecosystems and are either cycled through the ecosystem or transferred elsewhere. One of these cycles refers to the important element nitrogen. So let's look at three different parts of the nitrogen cycle with one part on this slide. And this involves a group of bacteria called nitrogen-fixing bacteria. This includes the photosynthetic cyanobacteria, a group of plant bacteria that form very close relationships with plants, and they're called plant symbiotes, different species belonging to the genus Clostridium and others. These are the nitrogen-fixing bacteria, which are, well, they used to be the only organisms capable of fixing molecular nitrogen, but now they know that some of the archaeans do this also. So what do we mean when we say that these bacteria can fix molecular nitrogen? That is, they can take nitrogen from the air, atmospheric nitrogen, and they can fix it. What does this actually mean? Can anyone tell me? Nitrogen fixation. Any idea?
So you're producing ammonium ions, so I guess
Yes, yeah.
Can combine with things like nitrates and
yeah. So essentially, fixing nitrogen refers to the ability to take gaseous nitrogen and convert it into a state that can then be used by those organisms and by other organisms also. So let's look at the reaction here. Nitrogen-fixing bacteria will take gaseous nitrogen or molecular nitrogen or atmospheric nitrogen, if you like, N2, couple this to 8 protons and 6 electrons, and this will form the ammonium ion. 2 ammonium ions, NH4+. So this is one of the important polyatomic ions that was introduced to you very early on in, I believe, lecture 3, the ammonium ion. Okay, why is this important? Why is the presence of the ammonium ion important in soil, for example, in soil solution? Because it's one of the only forms of nitrogen that plant roots can take up. Plants can't use atmospheric nitrogen at all. they need to absorb their nitrogen from the soil solution, and this is one of the forms that they can absorb. Okay, this particular reaction here is sensitive to oxygen. So it tends to occur in specialised cells that are virtually impermeable to atmospheric oxygen. All right, what happens once nitrogen is fixed into ammonium ions? That ammonium ion is then incorporated into certain amino acids, for example, glutamine and glutamate in the bacterial cell. And then in that form, the nitrogen or those amino acids can be converted into other nitrogen-containing compounds. All right, let's look at another part of the nitrogen cycle, and this involves various bacterial species called nitrifying bacteria. Nitrifying bacteria will convert the ammonium ion to nitrite. And some bacteria that do this belong to the genus Nitrosomonas. Okay, nitrite. Who knows the chemical formula for nitrite?
NO2 minus?
Yep, correct. Yep, so another one of our polyatomic ions, nitrite, NO2 minus. Can plant roots absorb nitrite?
Don't think so.
No, they cannot. All right, so that's not good for plants, but the next part is certainly good. Another group of bacteria belonging to the genus Nitrobacter will take that nitrite and convert it to nitrate. Who knows the chemical formula for nitrate?
NO3 minus.
Correct. So another important one for everyone. NO3 minus. Can plants take up nitrate from soil solution?
I think they can, yeah.
Yes, they can. So plant roots love ammonium ions and they love nitrates. So this is definitely a very good part of the nitrogen cycle.
Ammonium nitrate's a really good fertiliser, isn't it?
Oh, absolutely. Absolutely. All right, next part of the nitrogen cycle, and this is probably not a very good part of the cycle. This involves the denitrifying bacteria, including members of the genus Pseudomonas and various anaerobic bacteria. They will use that nitrite and the nitrate as terminal electron receptors or acceptors. So does anyone remember the electron transport chain that we mentioned in one of our earlier lectures? Yes, no?
Is that the ATP thing?
Yeah, absolutely. That's right. So terminal electron acceptors, the final terminal electron acceptor is usually oxygen. Well, that's what we use anyway. We use oxygen to basically scavenge those electrons and keep the electron transport chain going. Okay, these denitrifying bacteria don't use oxygen. They will use nitrite and nitrate. And as a result, those important nitrogen species in the soil solution will be converted to gaseous nitrous oxide and molecular nitrogen N2, and once again the nitrogen is lost from the soil. So that nitrogen is no longer available for other soil organisms or for plants, well, apart from nitrogen fixing organisms. Okay. All right, very briefly, some commercial applications of bacterial fermentations. And I'm sure viticulture or winemaking students can add an important one to this. But maybe not so much from bacterial fermentation, more from yeast fermentations. Okay, fermentation produces a range of end products, many of which are used in agriculture and food and alcohol production. Just a couple of examples shown here. Everyone's familiar with lactobacillus, lactococcus, and other bacteria that are used to produce yogurt and milk. Various species of bacteria bacteria are actually used to produce ethanol. So this is not the ethanol that you find in your wine, but commercially produced ethanol. Huge business. Another group of important bacteria, the methane-producing bacteria, also referred to as methanogens. And the methanogens will use hydrogen and carbon dioxide to produce methane gas. Obviously very important because methane gas is one of those important greenhouse gases thought to contribute to global warming. Methanogens occur in anaerobic environments such as animal intestines, waterlogged soils, and so on. All right, moving on now to our next kingdom, kingdom fungi, which includes yeasts, truffles, mushrooms, rusts, molds, and so on. Some characteristics of the fungi. They are all eukaryotic organisms. They are all heterotrophs. Many of them are filamentous. We'll come back to that. And they have chitin in their cell walls. Very tough substance, chitin. All right, here's some examples. We can see this disease here, which is caused by a rust fungus and a mushroom, another type of fungus. All right, classification is very complex. We can see some different phyla listed here. You don't need to remember these. These particular phyla here include those fungi that can reproduce both sexually and asexually. This includes the water molds, the sac fungi, and the club fungi. And the club fungus, called Armillaria, is pictured here, growing out of the trunk of this rotting tree. And what we see here, these are just the fruiting bodies of the fungus. Most of the fungus is actually found within the actual tree itself, within the trunk. Here we have a phylum that exhibits asexual reproduction only. Well, they've only observed asexual reproduction in the Deuteromycota. And here we have the lichens, which belong to phylum Mycophycota. And the lichen, of course, includes both a fungus and a photosynthetic organisms like algae. And they live very close together in this symbiotic relationship. All right, the fungi are the closest relatives of kingdom Animalia. So they share a lot of similarities, but here are a couple of differences. Fungi contain chitin in their cell walls. Animal cells lack that substance called chitin. Fungi have haploid nuclei in their non-sexual cells. What do we mean by haploid? We mean only one copy of each chromosome within the nucleus, whereas animals have what are called diploid nuclei, that is 2 copies of each chromosome in their non-sexual cells, otherwise referred to as somatic cells. Structure of fungi. Many fungi produce filamentous hyphae, singular hypha, and hyphae are simply very elongated cells, which look like very fine tubes. The outer cell wall encloses the plasma membrane, which in turn encloses the cytosol and all the organelles found within the cytosol, including the nucleus and the endoplasmic reticulum and all the other organelles that you would normally find within animal cells, for example. These filamentous hyphae elongate and branch at the very tips where the wall is still forming. So it's still quite flexible at the end of each hypha. Okay, we've got a great image here. Here's a spore, a fungal spore that has germinated. So the cell or cells that are found within that spore start to elongate to form these filamentous hyphae. And this spore has germinated on the surface of a plant. Okay, here's another hypha, and another one here. Now, this one has actually found an entry point to the plant tissue. So then it starts to form this infection structure called an aprosorium. Okay, the main body of the fungus is called a mycelium, and this basically just consists of a tangled mass of hyphae. All right, let's look at hyphal tip growth and absorption of nutrients. All right, so the very tip of a hypha, we can see some of the organelles, Golgi apparatus, and all those little vesicles that are budding off from the Golgi. Two major types of vesicles are pictured here. These little green ones, which are carrying cell wall components. And let's see where they're heading. So if we follow these green ones here, here's a vesicle that's fused with the plasma membrane. It deposits its contents into this region between the plasma membrane and the cell wall, and those vesicle contents are incorporated into that newly formed cell wall here on the outside. Then we have another group of vesicles which are carrying digestive enzymes. So this is how the fungus actually absorbs nutrients from the environment. It secretes enzymes into the immediate environment at the very tip. So that vesicle, once again, has fused with the plasma membrane, releases those enzymes, and those enzymes will break down large compounds. So, for example, large carbohydrates into monosaccharides such as glucose, and then the fungus can actually absorb those smaller compounds. So that's hyphal tip growth. And we can see that outer wall, which contains chitin, and then the inner plasma membrane. So as I said, a fungal hypha is simply a very elongated cell. It keeps growing at the tip, and now and then it'll branch off to produce new hyphae. Okay, fungal nutrition. Fungi digest nearly any form of organic carbon, which is a good thing. They're one of our major groups of decomposers. And fungi, in fact, are the only group of organisms that are known to digest lignin. So you remember that compound, that phenolic compound called lignin, which plants form, particularly in secondary cell walls. Fungi can actually break down lignin. Fungi are heterotrophs. They all seem to have a preference for glucose as their source of carbon, but they can basically use anything. They are all saprophytes, which means that they tend to feed on dead and decomposing material. As I said, they are important decomposers. They release those enzymes onto the substrate in their immediate environment, break it down, and then absorb those nutrients. Some fungi are highly specialised as parasites. Fungal reproduction. Fungi reproduce by making spores. And within a spore, you can find one cell or several cells that will eventually germinate. That is, those cells will start elongating when the spore germinates to produce fungal hyphae. If it's a filamentous type of fungus, that is. Changes in environmental conditions will trigger sporulation, that is the formation of spores and release of those spores from the fungus. And we can see a fungus up here that has started to produce spores. So all that black region there is the production of fungal spores on the tips of these hyphae. Down here, we can see some spores in more detail. These types of spores are called cnidiospores, and each fungal species will produce very characteristic shape of spores and the way that they're actually formed. And that's very important in terms of classification of fungi. So you can see these upright upright collections of spores, and the hyphae are down here. All right, many of the fungi will produce both sexual spores and asexual spores. Sexual spores, there's normally only one type of sexual spore that's produced by a fungal species. Asexual spores, there may be up to 4 different types of asexual spores produced by each fungal species. And viticulture students will be looking at this photo here in horror. Am I right?
Yep.
Botrytis fungus, yeah, in grapes. Okay, fungi and humans. Fungi obviously have an enormous impact on humans in terms of food and alcohol production. They can produce some pretty nasty toxins. You may have heard of ergots. They produce some important medicinal drugs. They cause some diseases of animals. They cause lots of diseases of plants. There are some fungi, for example, a particular species of rust that are being studied at the moment as biological control agents of weeds. So that's an interesting application. And of course, fungi are important in industrial mycology, production of various food acids and antibiotics. All right, another couple of nice photos. So here we have the hallucinogenic mushroom called psilocybe, used as a recreational drug. And here we have the fungus called Trichophyton rubrum, which causes that nasty ringworm disease. Moving on to our next kingdom, Kingdom Protista. I would have to say that just about every organism that doesn't fit into the other kingdoms have been thrown into Kingdom Protista. They are so diverse. The organisms found in this kingdom, some of them are actually more similar to plants, some are more similar to fungi, some are even more similar to animals, but they don't quite fit in those other kingdoms. So there's a lot of classification and reclassification going on in Kingdom Protista. Okay, what do they have in common? They are all eukaryotes, so even those that are unicellular and microscopic, they still are eukaryotes. Mostly they are unicellular and aquatic, but remember that there are some that are quite large and multicellular. Many of them have these little hair-like structures called flagella or cilia that they use to move around. Nutrition is very diverse. Some can photosynthesize, others are parasites, others are predators, and still others absorb their nutrients. All right, classification. Here are just some different groups of the protists, and you don't have to remember these. The choanoflagellates are marine protists. Then we have the slime molds. We've got photosynthetic protists, for example, red algae and green algae. The chromists, which includes diatoms, brown algae, water molds, and downy mildews. And viticulture students are saying, oh no, not the mildews. Alveolates, for example, the dreaded malaria parasite, and others. Okay, very interesting little life cycle shown here from your textbook. And this is a particular species of slime mold called Dictyostelium discoideum. And it's interesting because they live most of their life as single-celled amoebae. They're quite ubiquitous. They're found in moist soil, moist leaf litter. Very, very common, and they move around and they feed mostly on bacteria. But then when environmental conditions are not ideal or they're running out of food, they actually secrete chemical signals which basically makes them all congregate together into a mass, and that mass then forms these little slugs. They're about 2 millimeters in length. So each of these slugs contains thousands of cells, all aggregated together. This is what the actual slug looks like. This is an actual photo of a slug, but it's been coloured. And those little slugs move off until they find a suitable place. And then within the slug, some of the cells will start forming a flattened base. Other cells will form this stalk, and still other cells within each slug will form this fruiting body. And then within the fruiting body, cells will start to differentiate to form spores that are eventually released. So it's very interesting that that many of these cells basically sacrifice themselves because the rest of the cells here will actually die once the spores are released. And also interesting that you have these single-celled organisms that can come together, they communicate so well, come together and act like a multicellular organism does. So quite interesting there. If you're interested in actually seeing them at work, just YouTube, Google YouTube Dictyostelium discoideum. Cellular slime molds, really interesting. All right, some examples of algae. Here we have some nice photos of red algae, green algae, multicellular green algae. Down here we've got some single-celled green algae. A very large brown algae here, and also here on the right. So they're also all belong to kingdom Protista. Very briefly, let's look at biotechnology of green algae. This is an enormous area of research, or it's become enormous over the past 2 decades or so, and only some examples are shown here. So this species of green algae, Dunaliella salina, is extensively farmed for beta-carotene, which is a precursor of vitamin A. This is an enormous algal farm found in Western Australia. You can see all those ponds there where they farm Dunaliella. Another example, Haematococcus pluvialis is farmed for a substance called astaxanthin, which is used in aquaculture as a fish food additive. I think Darren and Brad should be presenting this slide. What do you think, guys?
Maybe next semester.
Next, all right. Batriococcus braunii. This is a really interesting example. This green algae actually produces these long chain hydrocarbons, very similar to what you would find in crude oil. So they're actually looking at using these hydrocarbons as an alternative fuel source. Fascinating. All right, moving on to our next group of protists, the oomycetes, which includes water moulds and downy mildews. These are very, very similar to fungi, but the reason they're not in kingdom fungi is that their cell walls contain cellulose instead of chitin. So you can see the problem here sometimes with classification. 2 examples of the oomycetes are shown here. Phytophthora cinnamoni, which causes extensive dieback of trees, and Phytophthora infestans, which caused the famous Irish potato famine in the 1840s. Okay, next group of protists, the alveolates. One group of alveolates is referred to as the dinoflagellates, and these are photosynthetic alveolates, commonly referred to as plankton. Okay, some species cause what are called red tides, and some of these are harmless, but sometimes these alveolates produce very, very potent toxins. This explosive growth here is due to high temperatures and excess nutrients. So where might these excess nutrients come from? From any waste that's going into the water, sewage waste, runoff from fertilised farms, and so on. All right, and I think this is our final example of a protist, quite a famous one, the protist Plasmodium falciparum. This is the causative agent of the disease malaria. Plasmodium has two hosts in its life cycle. It completes the sexual part of its life cycle in this mosquito called the Anopheles mosquito, and it completes the asexual part of its life cycle in liver cells and blood cells of humans. So in the actual mosquito, the Plasmodium tends to be found in very high concentrations in the salivary glands. So when the female mosquito takes a blood meal from a human, it's very easy to transmit these protists into the actual human. So that's Plasmodium falciparum. Okay, and we're nearly done. So we're just going to very briefly consider a group of entities, and you might wonder why I'm saying entities, because viruses are not considered to be living organisms. This goes back to our cell theory, which states that all living organisms are made of cells and the products of those cells. And herein lies the problem. Viruses are not made of cells. So at the moment, they're not considered to be living organisms, but there is still a lot of debate about that. So obviously, cell theory, that's something that we came up with to try to describe what it means to be living. So are viruses living organisms or not? All right, so how do we refer to viruses? We can call them small infectious particles or small infectious entities, but they are not at the moment classified as living organisms, so they don't belong to any of those kingdoms of life that we've been looking at. All right, they're not considered to be living because they have an acellular structure. They're not made of cells. They cannot reproduce independently at all. And they contain either DNA or RNA, but not both. Every other living organism contains DNA and various types of RNA. Okay, viruses tend to have one or the other. All right, let's finish with the structure of viruses. So all viruses are placed into one of two groups based on structure. This is one structure here. These are the naked virions. And the second group are the enveloped virions. Now let's just consider that term virion. So viruses in their complete form and the way that they're actually found in the environment are referred to as virions. But when one of these virions invades or infects the cell of a living organism, it's actually just the genetic material that infects that cell. And in that form, they're referred to as viruses. So when you have a virus infection, you actually have the genetic material of the virus in your cell. Okay, so two sort of distinctions there. But the terms virus and virion are often used interchangeably. All right, so let's look at these two different structures. All right, so this is the naked virion, and it consists of the nucleic acid to the inside. So this could be one or many pieces of DNA or RNA. And then surrounding the nucleic acid of the virion is a protein coat, which is called a capsid. And that's it. That's a naked virion. So no cell. Here we have what's called an enveloped virion. Once again, it contains a nucleic acid enclosed within this protein coat, and the protein coat often takes on this shape, the icosahedron shape. And then to the outside of that protein coat, we have a membrane. This is a lipid bilayer, which encloses, which surrounds that protein coat. So you might say, well, hang on, if there's a lipid bilayer membrane, why is that not considered a cell. And that's because this virion has hijacked this membrane from the last cell that it infected. It's taken a little bit of the plasma membrane of that cell, or it's taken a little bit of the membrane from the endoplasmic reticulum or from the nucleus. So it's basically stolen this membrane from a cell. But these little spikes that you can see here, these are virus-encoded proteins. These are not the proteins that were in that original membrane lipid bilayer. They're encoded by the nucleic acid of the virus itself. All right, so they're the two major structures that you will see. And here, just for your interest, is a very common rod-shaped virion. And that completes our lectures for tonight. We will finish the second part of this lecture next week, but let's look at some tutorial questions. Here are the last set of tutorial questions for Living Systems 1. And of course, they are not assessed, but of course, they're still important for the final exam. All right, before you all go, I'd just like to point out something that's quite important for one of your prac reports. So this is part of the subject outline here. And let me just move this up. Lab reports. Okay, so here's the different sections in that you need to include in your lab reports, or rather in, here we go, this is what I wanted to point out. So this reflection part of the lab report is required only for the acid-base titration report, and the reflection only has to be a very brief paragraph. And I'll just show you where the reflection is. So here's the marking template for the acid-base titration prac. And it includes all these grad caps. Here we go. So this is the part that relates to other reflection. Okay, so reflects on conclusion and validity of responses in relation to analytical approaches or feedback. In other words, you can reflect on any part of that prac that you like. You could reflect on how valid your results are. You could reflect on the information that was provided in regards to that prac, or you can reflect on any additional questions that you may have been left with at the end of the prac. So this is just a personal reflection. And as I said, you only need to write a very brief paragraph for that part of the prac report. Okay.
Savannah?
Yes.
As a reflection,
Yeah.
in the first tense, or is it still kind of the first person, sorry? Or is it still...
Yes, you can write in the first person for this. Yes. In contrast to the rest of the report, where you always have to write in the third person. Yeah, very old convention, that one. I think that's actually very slowly changing. I'm seeing more and more journal articles where the authors do say we. You know, that was never allowed in the past. But nevertheless, we still try to say don't write in the first or second person. So that's it for tonight. Who has any questions for me? Oh, these grad caps, my goodness. Marking is going to be a nightmare with all these grad caps. Look at that. Not every subject has these grad caps incorporated into them.
What are they, Savannah?
Oh, they're basically just, you know, they look at all these different, well, many of them look at generic skills that you normally pick up during any degree. but they've never been formally assessed in the past. And about 2 years ago, La Trobe and probably all the universities started formally assessing them. So here are some generic skills: interpersonal skills, participation, reflection, outcome orientation, independence, and so on.
Are they softer skills, if you like, rather than...
Sorry?
Are they softer skills, more about character and behaviour?
Yeah, absolutely, absolutely. And there's a whole suite of these graduate capabilities, which are always informally assessed, I suppose you could say, but now it has to actually be documented. So the way that they're assessed is that you are given either standard exceeded, so this is in regards to referencing, standard exceeded, standard met, or standard not met. So you're not given a mark as such for the graduate capabilities, but you are given one of these levels is assigned to each one. So many of these I'll be assessing as based on your report itself. So for example, referencing, the grad cap on referencing, I can actually take that from your assessed referencing in your report. So for example, if you achieved an excellent or very good for referencing, then I would give you a standard exceeded for that. If you got good for referencing, then I'll give you a standard met, and so on.
I'll tell you what it's like.
Yes, sorry?
I'll tell you what it's like. It's like writing year-end evaluations for staff, and based on 360 feedback. This is also a very similar grid, actually.
Oh, really?
Determining if someone gets promoted or not. Yeah.
Ah, yes, of course. Yes. So you'd be looking at all the different criteria.
Yeah.
That have to, yeah, absolutely, same sort of thing. So what you'll find as you progress through the course is that there are a couple of subjects in second year that have grad caps incorporated into the assessment, and also in third year as well. Yeah, the same grad caps, but different levels of competency, you could say. So obviously the grad caps in third year are a lot more advanced than these ones in first year, but they're assessed in the same way. So they're graduate capabilities, making our job a lot harder, I think.
A lot more admin.
Yes, more admin, absolutely. More admin. Absolutely.
Silvana, on the lecture that we've just done,
yes.
Notice that it covers a lot of different chapters in the textbook. Is it really just focus on what was covered in the slides rather than...
Absolutely. Yes. No, don't go to the textbook. There is so much information given in all those various chapters. Just look at what's in the slides, and in particular, focus on the tutorial questions for this particular lecture. Absolutely. So next week we'll finish lecture 18, and then we'll have a bit of a discussion about the final exam and what you should focus on. Okay.
Well, I'll see you in full-time class next week.
Oh, will you?
Yeah. Yep.
Oh, terrific.
Yep.
Yeah, terrific.
Yep. Okay.
Because I haven't had many turn up the last couple of weeks.
Oh, okay.
Which is pretty standard at the end of semester.
Well, that's when it gets harder, though.
Yeah, well, that's when everyone's rushing to complete a million assessments. Absolutely, yeah, that's pretty standard at the end of every semester. Attendance really, really drops off. Absolutely.
And another sort of just practical question, because I saw the exam timetable's out.
Yes, it is.
No concept of where the exam actually is taking place.
Oh, right. It doesn't say on the timetable?
Well, it says, but it's just a code, so I didn't
Oh,
understand
yes.
where that meant.
Don't worry about that. I'll be putting up a campus map of the Preston Mel Poly campus. So once I get that organised and put that on Moodle, then I can show you exactly which building to go to find the room where your exam is taking place.
Okay, but it's at Preston, not Epping.
Yeah, definitely at Preston. Yep, all the exams are at Preston, as far as I can see. Yeah, so we'll have a look at the Preston campus map next week.
Okay.
Okay,
Actually,
so
I will be doing this at night. I just remembered I've got a tractor license, but it was supposed to start on Tuesday, but it's actually starting on Monday now.
Oh, that's right. Yeah, the tractor course.
That's right. Yeah.
Yeah, a few students told me today that they wouldn't be in next Monday because of the course.
Yeah.
Yep, that's fine.
Yep. Okay. Well.
No problem.
To this then.
I'll see you in the evening then.
Yep. Yeah. All right.
All right, that's great. Thank you, everyone.
No worries. Have a good week.
For your wonderful attention, as always.
Thanks, Lavon.
Thank you, everyone. Have a great week.
Bye.
All right, bye-bye.